Okay,
And
the recording is starting.
okay, so tonight for our IABDM Zoom meeting, we have Blanche Grube, who is the owner of DNA Connections, and she is going to explain to us the value in sending off your teeth, your pathology reports to DNA Connections to validate what in the world is going on inside of these teeth. We need more validation so it's not just hearsay that it was a bad tooth. But then, even more importantly, once you get the report back, what in the hell do you do with it? Take it away, Blanche.
Thank you very much, Dawn. And the timing couldn't be any better because every night we're hearing on TV that they're doing tests for COVID-19. And some people are saying, well, you know, we don't have enough time to do a DNA test or an RNA test. Maybe we'll just do an antibody test. And so all the things that we do at our laboratory on a daily basis is actually being discussed by good old Uncle Donnie on TV. And I'm loving it. I really am. Great. Okay, so now let's go to my next slide. First slide, of course, is the title, Advancing Whole Body Health Through DNA Testing. So I'm going to go through the first few slides very quickly because this is just a refresher for the dentist or medical doctor who has not opened up an article or a textbook since school. And I know I'm preaching to the choir here. That's not our crowd. Our crowd are people who are constantly reading and staying up on top of everything. But just so that it's a congruent slide presentation, I'm going to read a little bit about what is DNA, just so that you have it fresh in your mind, because you know, a lot of people just forget a lot of things. So DNA, deoxyribonucleic acid, is a hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Dr. Huggins used to call it our address, our name and our address, or our license plate. He used to love using that term. Most DNA is located in the cell nucleus, where it's called nuclear DNA. But a small amount of DNA can be found in the mitochondria of each cell, where it's called mitochondrial DNA. And it's the mitochondrial DNA that have allowed researchers to trace back all of human ancestry to what they call the mitochondrial DNA woman who lived in South Central Africa. And supposedly the whole human race comes from this one woman. It's the mitochondrial DNA that they've been able to trace. Okay, and so of course everybody knows mitochondria is where we get our energy from. Again, the basic structure of DNA, which a lot of people forget, is really just simply a series of proteins. And the series of proteins all have a partner, and the partnership of these proteins is consistent. And it is just absolutely phenomenal to me that this series of protein molecules determines what kind of enzymes the body can make. And then because it determines what kind of enzymes, it also determines what kind of biological chemical reactions we're going to have in the body. Such a simple thing like one adenine next to a thiamine, or here's a guanine and its cytosine is missing. It's only those 4 different proteins. I mean, it's just phenomenal. If you just take a second and think about it. 4 proteins determine how you look. And there's no 2 people on the planet, we're close to 8 billion now. There's no 2 people of those 8 billion that look exactly alike. It's just mind-blowing to me. It really is, if you think about it for just a second. The four different proteins, and in their order, determine what you look like, how tall you are, what color eyes you have, what some of your personality traits, how healthy or not healthy you are. Amazing. Absolutely amazing. So basically, DNA makes you what you are. Okay, so how can analyzing DNA help to detect disease? Well, all diseases, every single one of them, have some sort of genetic component. And understanding the genetic mechanism behind these diseases helps to develop early diagnostic tests, new treatments, and or interventions to prevent onset or minimize severity. Now, mutations happen. I'm not an evolutionist, so I don't believe that mutations happen and they actually make the human being or the animal better. I don't believe that we mutated from a single cell mitochondrial type cell and then became a single cell organism, and then we became an amoeba, and then we became a little guppy floating around in the water, and then that little guppy became a fish, and then somehow or other the fish disappeared, and that guppy became a frog, and then it went from frog to, you know, all kinds of vertebrates on the planet, and then it turned into a bird, and then some of those birds turned into bats, and some of those bats turned into mammals, and before you know it, we had a human being. Really? I've always understood mutations to be more of an accidental thing. They come from the environment. It's what gives us not only our gene makeup, but our gene expression. How we express our genes is determined by our environment. And it's usually a turning off of good expressions. It's usually a turning off of good things. So I'm sorry, I tend to get off topic when I speak, and I get into my whole emotional rant and rage. So mutations may be inherited or developed in your time in response to environmental stresses, such as viruses or toxins or chemicals. You know, I think if you could take a look at the genetic makeup of our population, say the people who are in their 60s today, their genetic makeup is not the same as it was when they were in their 20s. It has changed, and unfortunately, it has not changed for the better. We are not evolving, we are devolving. Of course, the ultimate goal in using all this, getting all this information, is to help people, to make life on this planet better. So, what kind of genetic testing is there out there? There's several different methods, and I'm always kind of amused when people come up to me and they say, "Hey, Blanche, can I send my blood to your lab and, you know, find out about my personality?" I say, nah, you can't do that. That's not the kind of genetic testing we do. There are 3 types of genetic testing: cytogenetic, biochemical, and molecular testing. These are to detect abnormalities in the chromosome structure itself, how, once that chromosome determines it's going to make a protein, how is that protein functioning or not functioning, or the DNA sequence itself. Okay, for small DNA mutations, usually direct DNA testing is the most effective method, particularly if the function of the protein is not known, and a biochemical test has not yet been developed. A DNA test can be performed on a variety of biological samples and requires very small amounts of sampling. Those of us who are old enough to know that there was once a famous murder case, and there was a famous movie star. Was he also a basketball player? I don't remember. And, you know, it was the DNA testing that determined whether or not he had committed the murder of his wife. So it's people in those days used to make jokes about him traveling to West Virginia, because in West Virginia, everybody's DNA is the same. It's supposed to be a joke. Okay, so for some genetic diseases, though, multiple different mutations or variations can occur in the same gene and then result in the disease, making molecular testing very challenging. So DNA Connections, that's the laboratory that we own in Colorado Springs, Colorado. We are a biosafety level 2 CLIA certified laboratory. And we encompass the following specialties. We do bacteriology, we look at mycology, we look at virology, and the toxicity of microorganisms, which is a very important subject for dentists. And the technique that we do at DNA Connections is called polymerase chain reaction, PCR. So up until the COVID virus came along, Nobody knew what PCR was. And if I told somebody, well, we basically do PCR testing, they just gave me that deer in the road look. What the heck is PCR testing? Well, it's sometimes called molecular photocopying. The polymerase chain reaction is a technique used to amplify or copy or make a lot more of a very, very small amount of DNA. Somebody can give us literally a pinhead of DNA, and we can multiply it, make copies of it via PCR. We can make copies of it and then make more copies of it and more copies of it until we have enough copies that we can actually analyze it and study it. We could not do that until we had PCR amplification testing. So PCR testing is a direct testing method, as opposed to an indirect testing method that some people are very familiar with, a company called IGenix. Now, what they do is indirect testing, meaning that they're looking at the body's response to a microorganism. When the body gets a microorganism in it, it develops antibodies against that microorganism because that's the function of the immune system. The immune system cells spend their time floating around the bloodstream, looking inside some of the tissues and determining whether something is self or not self or not self. And the moment the immune system, one of these white blood cells, recognizes not self, it will flag it. That's specifically, for those of you who are interested, that's specifically the lymphocyte. It will flag that non-self protein, antigen, whole cell, virus, whatever it is, it will flag it and then alert the rest of the immune system to come and take care of it. Well, there are patients who have had, let's give an example, Epstein-Barr. They've had Epstein-Barr for 40 years. When you get to the point where the patient has had something for that length of time, when you go to test them, they may actually wind up having very few, if any, antibodies to Epstein-Barr, even though they've had it in their body for a long, long time. The body actually gets tired of putting up a front and not winning that front. So the body then kind of just moves on and pays attention to something else. And that patient will have very few antibodies, even though they have Epstein-Barr virus in their system. They've had it for 40 years, but they're not putting up a good fight. And because they're not putting up a good fight, they're going to have very few antibodies. And so when you do an indirect test like that, you might actually come up with the false conclusion that this patient doesn't have Epstein-Barr, when in fact, they do. PCR, on the other hand, is a direct method testing. So we're not looking at the patient's immune response. We're not looking to see, where are you in this whole episode of being contaminated and putting up a fight, and then either winning the fight or not winning the fight, and your immune system gets tired. We're not interested in that. We're interested in, do you have the footprints of the microorganisms in your body? And of course, the footprints is the DNA. If the bugs were there, they will leave, if not all of their DNA, they will leave parts of their DNA. That's the part we test, and we can tell if it's been there or not. You know, it's kind of like looking inside my son's kitchen. Sometimes I can't tell if he's had a party there or not. But if I walk into his kitchen and I see tons of beer bottles on the kitchen table, it's a pretty safe bet that not only was he there and he had some friends over, but one of his specific friends that drinks an awful lot of beer, I know he was there because it's the evidence is there, okay? So it detects, direct testing tells you presence or absence. And it's also incredibly species specific, which means we're not going to tell you, oh, there's a whole bunch of microorganisms that have these characteristics, and yeah, some of them could be there, maybe some of them not. No, we're going to tell you specifically exactly which species was there. And then, of course, it's up to the doctor to target the treatment. One of our frustrations is that we are a laboratory. We're not a diagnostic clinic. And so when the doctors call us and say, well, my patient has these microorganisms, how do I treat it? Well, we kind of scratch our head and go, wait a minute. We're the laboratory that gives you the name of the microorganism. You're the doctor that's supposed to know how to treat it. And so I know it's very frustrating for a lot of doctors, because the last time they looked at microorganisms was when they took the boards and got out of medical or dental school. And so I understand very much that it's very frustrating for doctors to not know how to treat these things. But no matter how much we try to help you, it's still going to be the responsibility of the doctor when treating the patient, not ours. So how does PCR work? Well, first the sample is heated. And when the sample heats, the DNA denatures or separates. Instead of having that double coiled helix, it separates into 2 single strands of DNA. Then we add to that test tube, we add primers. And it's a proprietary chemical that we're not going to tell you what it is. But it takes those denatured strands and anneals the other proteins in the solution to those denatured strands. The next procedure is to add an enzyme called Taq polymerase, which builds 2 brand new strands of DNA. So you started out with 1 strand, it splits in half with the heat, then the enzymes and the Taq polymerase add the proper proteins to the proper ones on the strand already, and now we've got 2 strands of DNA. Then the process is duplicated again with the same original DNA, and now we've got 4. And this is how we make more and more copies. The whole process is repeated 30 to 40 times, leading to 1 billion exact copies of the original DNA segment. So this kind of gives you a visual of a sideways pyramid. You start with the template DNA, then you go to the first cycle, you've got 4 copies. Then you go to the second cycle, you've got 8 copies, and then 16, then 32, then 64, and on and on and on. And by the time you've done this 30 or 40 times, you've got a lot of copies of that DNA. So that's called amplification. Once the DNA is amplified and produced by the PCR technique, you can do all kinds of experiments on it. For example, the Human Genome Project was finished years and years ahead of its scheduled time because of PCR, all right? There's such a thing as PCR fingerprinting, detection of bacteria or viruses, and diagnosis of genetic disorders. I won't spend a lot of time talking about genetic disorders because everybody really wants to know, what do we do with the microorganisms in root canals and cavitations? But one of the genetic disorders that we do test for is for gluten intolerance. And here again, is it a disorder, or is it an improvement over the genome? I'm gluten intolerant, so I would say it's a disorder. Doesn't make me happy that it's Friday night and Blanche does not have any real pizza. I mean, New York style pizza in front of her. Nope, can't do it. Cannot do it because I am gluten intolerant. So we have something called the oral panel. This is the one that is most used by dentists. And they will send us root canals, the blood from cavitations, blood from root canals. They'll send us superfloss. And the oral panel tests not only for bacteria, but viruses, parasites, and fungi. We test for 88 different microorganisms. And again, it is species specific. So give you just some examples. Heliobacter pylori. This is a very nasty microorganism that has now been shown to cause gastrointestinal cancer. Clostridium difficile causes colorectal cancer. Enterobacter SP, I forgot what SP stands for. Just had a senior moment. Causes sepsis. Fusiform bacteria, perio disease, and skin ulcers. Streptococcus, causing mild infections to pneumonia. You know, when most people come down with the COVID virus, they don't die of the COVID virus. What they die of is pneumonia. When people get the flu every year and they get pneumonia, they're more prone to getting pneumonia. And so that is what they die of. That's why I'm staying in Florida until it's nice and warm up north. Then maybe I'll go. Staphylococcus causes a variety of systemic diseases. And then, of course, there's the human papillomavirus 16, 18, and 58, and we test for all of these. These are the viruses that are most common in sexual transmitted diseases and in cervical cancer. This slide shows the different species, or the different specimens, that's what I meant to say, the different specimens that you can send. You can send us blood, you can send a piece of tissue, you can send teeth, you can send the whole root canal, you can send the goobers and the globby stuff that you get inside of a cavitation. That's the picture that you see on the lower left, that's a cavitation. You can send a section of bone graft. I can't tell you how many times I've opened up a cavitation and the bone graft material that the dentist put in there 10 years ago that was supposed to turn to bone did not. It's just sitting there. And sometimes it looks like the little room that they throw the kids in these rooms at Chuck E. Cheese, and these rooms are full of little plastic balls, and the kids have a great time jumping into these pools, these swimming pools filled with little plastic balls. Well, I've opened up many a cavitation and have found tons of little white balls. And I have no idea what kind of graft material it is, because that's not what I do. I don't play around with those things. But obviously when I go in there and scoop them out and wipe them on a piece of gauze, they're obviously not bone. It's little look like little plastic white balls that have been just sitting there, I guess waiting for some kid to jump in. I don't know. You can send us paper points when a patient has a periodontal pocket, or they have a root canal tooth and they really don't want to get it taken out unless you prove to them first that the tooth is toxic. So you can stick a piece of paper point in between the root canal tooth and inside the sulcus, send that in, and then we will tell you what kind of exudate is flowing from the inside of that tooth to the outside. And you can almost put money on it that you're not going to get back a report saying that that root canal tooth is just so healthy and fantastic. You can leave it there because it's just perfect. That's not going to happen. Okay. In the lower right hand picture, you see superfloss. And so we have some patients who will send us some superfloss that has been rubbed up next to a root canal tooth or any tooth that's in question. Okay. Okay, so again, what we test for is microbial presence. Is the DNA there of the microorganism, or is it not? And just how much? What's the quantity? So it's not simply presence or absence, but the brightness of the amplification product is directly correlated to the quantity of microbes present in the starting material. So the luminosity, in other words, how white is it? We put on a scale of 1 or almost 0 to 10. And so you can see the one on the left-hand side is a 10, and the one on the far right is a 0 or a 1. A very bright band, like the one on the left side, represents a starting amount of millions of microbes. That's right, millions. That's what I said. Even though we started out with a pinhead, by the time we finish amplifying it, we've got millions of them. And a faint band on the right side represents possibly 10 to 20 microbes. Now, anybody who knows anything about immunology is that if you have 10 to 20 microbes in the whole body, you probably have nothing to worry about. The immune system can take care of that. What your immune system cannot take care of is millions of microbes. So each microbe that we get is assigned a virulence risk factor. In other words, just how bad is this bug that you're telling me about? I can't even spell it, no less pronounce it, no less tell you how bad it's going to be. And that's the difficulty that the doctors are having. It is dependent on the microbe's innate biological risk. In other words, we look at how much involvement is in disease causation and severity. In other words, what kind of diseases does this bug cause? Does it only cause slight periodontal disease? Or does it cause periodontal disease that's so bad that the patient will have a heart attack? And what's its prevalence? And how resistant is it to antibiotics? Of course, the more resistant to antibiotics it is, the worse it is. And then, of course, there's the diagnostic and treatment difficulties, the presence or absence of inherent toxins. So a proprietary algorithm that we developed calculates the total risk factor, taking into account the overall virulence of the organism and the amount of the microbe present. So we look at both ends of it. Isn't that a pretty mouth? Wow. Now, there's no way on the planet a person with this kind of mouth could have a bad mouth, but a healthy body. It's just not going to happen. Okay? And our report includes the microbes detected with the total calculated risk factor, a generalized microbial description. So we're not going to make you microbiologists, but we do include a paragraph telling you what this microbe is famous for. And then we tell you what are the symptoms of infection. And so the patients can actually look at that report and say, oh yeah, you know what? I was getting palpitations of my heart. Or yeah, you know what? My stomach was burning every time I would try to eat something. And you begin to wonder, maybe they should go for a little bit more of a further workup with the medical doctor and find out, do they have stomach cancer? It's a good idea to do. And then, of course, we include also what the CDC recommends. I didn't say World Health Organization, I said CDC, because if it was the World Health Organization, we couldn't tell if they were telling the truth or not. And I promised I wasn't going to get political during this talk. There I went and got political. Okay, so let me give you some examples. Actinomyces nislundi has a total risk factor for a patient of 14.28. These are bugs that are gram positive. Most people know it's the gram negative ones you really have to worry about. But gram positive can be bad if there's tons of them. So they're normally present in the mouth. Okay, so all right, right now we're not really that concerned, are we? But they can cause infections in the mouth. What are the symptoms of infections? Well, it can actually form an abscess in the mouth. If it's there long enough, it can actually go down into the lungs, and then it can carry on into the gastrointestinal tract. So a little bit of Actinomyces species, not a bad thing. A lot of Actinomyces species, it's a bad thing. And these bugs are sensitive to penicillin. So here's where I myself, as the owner of the laboratory, as a biological dentist, as a doctor of integrative medicine, here's where I have a problem. Because I know from my training, if I'm going to give a patient penicillin for a mild case of actinomyces, I am literally trying to kill an ant with a frying pan. Not a good idea, because that penicillin is going to wipe out all of the good microorganisms in that person's intestines. And so as a doctor, you really have to weigh whether or not you're going to give the patient an antibiotic like penicillin, or are you going to try more natural treatments? Of course, in my office, I would be going for the more natural treatments, okay? When the patient has a penicillin allergy, we're looking at doxycycline, we're looking at tetracycline, we're looking at the alternatives to penicillin, all right? Again, you have to really weigh whether it's worth it to wipe out somebody's intestines to get rid of a mild case of Actinomyces nislundi. Okay, here's another example. Campylobacter shoei. Campylobacter shoei is a gram negative, so we automatically are now thinking to ourselves, this is a little bit more of a bad boy. Okay, and it's a microaerophilic spirochete, which means that if it finds an area where there's no oxygen in the mouth, it's actually very happy. And these spirochetes are the ones that you will see on a glass slide should you be a really good practitioner, good dental practitioner, and you take a sample of the fluid that is in between the tooth and the gums. You take a sample with a probe, you put it on a glass slide, and you stick it underneath a microscope. When you do that, you will see these spirochetes, and they look like little corkscrews. you will see them swimming across the glass slide. They're very motile. They're either unipolar or bipolar flagella, and these little guys go wherever they want to go. So when you see them in the mouth, you can be sure they're not staying in the mouth. These little spirochete critters will go directly from the mouth and try to get to the heart as fast as they can because they like the heart. It's nice, it's juicy, it's warm, it's a great place to multiply, and it's a great place to give the patient a heart attack. Of course, once they have the heart attack, then the spirochetes are going to feast for maybe a day or two, and then they're going to die because there's no more oxygen, no more food for them. Okay, so Campylobacter shoei produces inflammation, bloody diarrhea, dysentery, cramps, fever. So if you have it in your gut, and if you have it in your gut, it came from your mouth. If you have it in your gut, you're in serious trouble. Again, what is the treatment? What would we do in the biological world? Well, the first thing that comes to my mind is grapefruit seed extract. A couple of drops of grapefruit seed extract in a glass of water. That's the first thing that I would use. Conventional doctor, they're probably going to go with quinolone antibiotics, ciprofloxacin, levofloxacin. They're not effective though. There are too many of these bugs that are already resistant to those. So you really have to start thinking outside the box and look at more natural ways of killing these bugs. All right. Streptococcus intermedius. It's got a total risk factor of 16.83. It's a gram-positive bacteria. So, okay, maybe it's not so bad. It's commonly found in the mouth, but it does have a proclivity for abscess formation. So when you see Streptococcus intermedius, it's going to form an abscess, and if it's in an upper tooth, that abscess is going to go right to the brain. If it's in a lower tooth, it's more likely to go down to the liver. But it can cause endocarditis. Okay, infections are rare in adults with normal functioning immune system. So again, if you have a few of these bugs, your immune system can attack them in the mouth before they travel onto the rest of the body. If you don't have a good immune system, or your neighborhood has just turned 5G, you're going to have a problem dealing with this because the body can only handle so many stresses at one time. We can handle microorganisms, but if you add electromagnetic fields to our bodies, we're going to have a harder time handling microorganisms. Okay? Streptococcus intermedius is highly resistant to many antibiotics. It could give a rat's ass about penicillin, ampicillin, doesn't care. So are you going to give something strong like ceftriaxone? No. I would go with ozone treatments. I would go with grapeseed extract. Those are the kind of things that I would use. I would probably make sure that the patient had ozone treatment at both ends. In other words, in the ears, drinking ozone water, and then rectal insufflation of ozone. That's what I would do in the holistic world, okay? Oops, 2 slides, 1. Okay, so when you get this report, you look at it and you see red. And of course, the patient is the first one who's going to freak out. And then the doctor, who doesn't have any answers for the patient, the doctor's going to freak out also. But let me explain it to you. If you see it in blue, like Rothia dentocariosa in this particular chart, it has a virulence of 7.5 or less. That's not bad. If it's in purple, like the Streptococcus mitis you see at the top, and the Provotella nigricans, then that is a moderate risk of 7.5 to 9.0. That's something the patient would want to have to take care of. But the serious risks are those that are in red, and those are all over 9.0. Now, you may have a few microorganisms, but they have a very serious history of causing pathology. And so they gain points and they wind up being in the red. Okay, I hope you understand that. On average, we find 12 to 17 organisms in one sample. 12 to 17. This is an interesting slide here because this slide came from a gentleman who grew most of his own fruits and vegetables on the island in Hawaii. He was a healthy man. He was not obese. You know, didn't have any gum disease, was a non-smoker, non-drinker, really healthy man. But they decided to do super floss on him and look at some extracted root canal teeth, some dental implants he had, and cavitations, and look at the number of red above 9.0 microorganisms. It's the most we've ever seen in anybody. And on face appearance, he looked like a healthy guy. So what is it that we see most often? When we look at the generalized oral panel, we see, you can see, I don't know if you can see the small print, but on the left side, all the way on the left side, Actinomyces neslundi, we see quite frequently because these are normal bugs that you would see in everybody's mouth. Fusiform bacterium vincente, again, it's something that we see in everybody's mouth. Even though you see a brown bar here, if a person had fusiform bacterium vincente, which in the old days, in the 1950s, 60s, and 70s, we used to call that trench mouth. If they had a lot of those microorganisms, then you would see a red bar on your test results, okay? As you travel to the right side of this graph, you will see the microorganisms that we see seldom, but some of them, like Serratia liquefactions, that's a microorganism that actually eats up dead tissue. It shows itself when the body has died and has been dead for a couple of weeks. And it actually eats up dead tissue and leaves a pool, a little puddle behind. And we've actually seen that in people's mouths. That's pretty gross. That is really pretty gross. So total risk factors given for each microbe detected based on their prevalence, how often do we see it, the characteristics of this microorganism, what diseases does it cause, and what our different treatment options are for it. You know, is this a bug that is resistant to a lot of antibiotics, or is it a bug that's, you know, easily gotten rid of? Well, if it's easily gotten rid of, then why give an antibiotic? Why not just use salt water? Why not use ozone, okay? And its relationship to systemic diseases. This was what determines how much of a risk factor are these bugs that you see, and they have a red bar next to them. Okay, so I'm going to take a few minutes now to talk about one of our newest tests called the propensity panel. The propensity panel from DNA Connection identifies 19 of the most common species, not just what we find in periodontal disease, but those bugs that have been implicated in chronic and systemic conditions. Their ongoing presence and chronic inflammation in the body causes these microbes to lead to their release and spread throughout the body. So the whole world is starting to wake up to the fact that if you have serious gum disease, that is the #1 cause of cardiovascular disease. And I'm happy to say it's even above obesity, it's above high cholesterol, it's above high blood pressure. Periodontal disease is the winner. The microorganisms that we find in our propensity panel are the ones that cause cardiovascular disease. They've been implicated. In other words, when somebody has cardiovascular disease, sure enough, those bugs are there. When somebody has gastrointestinal cancer, sure enough, those bugs are there. And yes, I know there are some people out there, some so-called doctors, who will say association does not imply causation. Well, yes, I have to agree with them. Association does not always imply causation, but very often it does. So don't, like I always say when I'm giving lectures, don't go above your 8th grade mentality. If you see the bugs are always there, every time there's a disease, a specific disease, you've got to think there's a relationship between the two of them, okay? The inflammatory process allows these microbes to ultimately spread at the systemic level. Okay, so what did Blanche just say? When you have inflammation of the tissue, you have breakdown of the tissue cells. breakdown of the tissue cells allows these microbes to spread because many of them are actually inside the cells. Okay? So once it spreads and it goes throughout the body, then you have the cells that cause inflammation. What they're, everybody's talking about now is the cytokines. That people who have chronic inflammation in their body already have a tremendous amount of cytokines being released from their broken down cells, being released into the body. And then they come in contact with the COVID-19 microorganism, and they have what is called a cytokine or cytochrome storm. Why? Because they've already had a lot of cytokines floating around in their bloodstream. So these are the people, these are the sickest people who are dying of COVID-19. The ones that already have chronic inflammation in their body. They already have chronic lung problems. They already are suffering from obesity. Everybody who's obese has cytokines floating around in their bloodstream because obesity is a very painful disorder. Been there, done that, that's for sure. All right, so next slide. Then when you get the copy of the propensity panel, what we do is we give you the 19 microorganisms on the left side, highlight the ones that your patient has in a large number, or it's a low number, but it's a very, very virulent bacteria or microorganisms. We highlight those for you, and then we check off the categories that these bacteria have been implicated, okay, not convicted, implicated in either cardiovascular disease, metabolic health, diabetes, resistance, pregnancy, development of cancer, dementia and cognitive health, respiratory tract infections. I mean, that's very popular today. If a patient already has chronic respiratory problems, you want to take a look to see, are they housing these microorganisms in their mouth? Is it a breeding camp for these microorganisms? And is that what's feeding the respiratory tract infections? Okay. And so we hope that we've developed this chart so that it would be nice and simple and easy for the doctor to read. You can take a look at this and talk to the patients and tell them that, yes, we're not saying you have cardiovascular disease, but you're certainly at risk for it. I mean, there's plenty of research to verify and to substantiate what I'm saying, that these microorganisms are associated with these diseases, okay? So what it says on the bottom is exactly what I just said. The panel provides insight to the doctor and to the patient to improve prevention strategies regarding the genetic propensity of the patient's risk. In other words, what are the chances of them getting that disease? Okay. APOE is another test that we do. Apolipoprotein, APOE test, is a genetic assay that looks at the protein structure in your genes to tell you what your propensity is towards developing Alzheimer's disease, dementia, but it also tells you how good are you at getting rid of toxins. This protein combines with fats, lipids in the body to form molecules called lipoproteins. We get the lipo meaning fat and proteins. These lipoproteins are responsible for packaging cholesterol and other fats and carrying them through the bloodstream. Maintaining normal levels of cholesterol is essential for the prevention of disorders that affect the heart and blood vessels. I've talked enough to most of the members of IABDM, and I've talked to them about cholesterol, and it is probably the number one material in the body, fatty acid in the body, that is so misunderstood. People think, because the medical establishment has told them this 100 times, that having a cholesterol below 200 is a good thing. And I'm here to tell you that cholesterol above 200 is much better for you. Your body needs the cholesterol to prevent you from having a heart attack, to prevent you from having a stroke. It's the patients with low cholesterols that are the ones that are so incredibly toxic that their body is sucking up the cholesterol as fast as as the liver can make it, and as fast as they can eat it. People who have low cholesterol very often are so toxic they can't eat enough cholesterol. They cannot. They cannot eat enough cholesterol to get that cholesterol up above 200. Trust me, I've been trying for almost 30 years now to get my cholesterol up. It used to be as low as 129. That is the sign of an incredibly toxic dentist. I had such a low cholesterol that my body was constantly fighting all the toxins in my body and using up that cholesterol. So thank God my APOE number is 33, which means I fall into the category of 80% of the population, which means I do okay in getting rid of toxins. Not great, but okay. So if you have a deleterious mutation of the APOE gene, then it is usually linked to mercury exposure. So, I highly recommend all of the dentists, certainly, to have the APOE test and see where do you fall in. Are you in the 3-3 group and you're with 80% of the population and have a pretty good chance of detoxing with a lot of work? Or are you in the 3-4 group, or God forbid, in the 4-4 group? And you would have a very difficult time detoxifying and detoxing the mercury that you come in contact with every single day. I would venture to say that if a dentist turned out to be a 4-4 or a 3-4, I would highly recommend they start studying orthodontics or get out of the dental profession altogether, or as soon as they possibly can, because the dental profession is a very toxic profession and they don't do a good job of detoxifying. They really don't. And so it helps you to make, smart, healthy policy decisions. And it helps you to decide what you're going to do with your future. Okay, we also do the Lyme panel. And we've had dentists who have sent us samples that they wanted us to do the oral panel and let them know which of the 88 microorganisms are there. We've also had them say, oh, you know what, I also want you to do the Lyme panel. Because believe it or not, we have found the microorganisms that cause Lyme disease inside of a root canal. Okay? So with the DNA Connections Lyme test, we not only test for Borrelia burgdorferi, but we also test for the 10 common Lyme disease co-infectors. And that's what the test looks like. When you get the results back, we highlight the microorganisms that the patient has. Now, as dentists, I don't recommend you tackle something like this. I really don't. If you have a patient that has all of this, I recommend you, send them to not only a naturopath, but a medical doctor also. And hopefully the medical doctor is not a part of the doxycycline club. I hope that they know a little bit more than just giving the patient doxycycline. Okay. Let's see, how much time do I have? Oh boy, I only have 6 minutes left. Wow. Okay, when the patient, well, there are a lot of, basically, I just want to say there's a lot of similarities between Lyme disease and heavy mercury poisoning. There's a lot of similarities. As a matter of fact, there's probably about 26 symptoms that are common to Lyme disease and common to mercury toxicity. And the only ones that are not common is that in Lyme disease, you have a tick bite. In Lyme disease, you have the bullseye. And in Lyme disease, you start out with a fever and things like that. But otherwise, there's a lot of similarities between the two. So this is why it's important for dentists to understand Lyme disease and understand the patient that comes to them complaining of symptoms. Yes, they may also be heavy metal toxic, but they may also have Lyme disease.
Hey, Blanche.
Okay. So, yes?
Is there a lot that you have on Lyme? If so, do we need to make that a next week meeting?
No, I think I'm almost finished.
Okay, no worries. I just didn't want you to rush if there was a lot on Lyme.
Okay, no, I'm pretty much finished now.
Thank you.
Okay, so why do the testing? Okay, you got to do the testing so that you know what microorganisms are in that tooth. And unfortunately, we still have boards out there that think that if you took a root canal tooth out, you took out a perfectly healthy tooth. And, Dr. Huggins researched this for years and found out that, he was right. It is all in your head. And this world needs so many good biological dentists to get them out of the sewer hole that they're in from all of the very poor dentistry that was done in their mouth. And I don't mean poor technically done. Usually it's technically perfect. It's just that it's going to kill the patient slowly. So interpretation of the DNA test, we are not, again, I have to say this, we're not a clinical diagnostic laboratory. We can only give you the results of the DNA PCR testing. It's up to you doctors to take a look at it, to know something about it, but then to really to recommend another qualified healthcare provider for the diagnosis and the treatment. You don't want to turn your office into a doxycycline clinic, okay? And so, yes, now I am finished. Thank you. And do we have time for questions?
Yes.
Good. Any questions?
I think people need to unmute.
You have to unmute to ask a question.
Well, maybe you were just talking to the choir and you got a bunch of geniuses here.
I think so. I think so. Well, that's fantastic. If you're shy and you don't want to ask a question now, feel free to send an email to DNA Connections. You can send an email to Leslie, to our head PhD. She'll be happy to answer them. You can send an email to me, BDGrube at AOL, and we'll be happy to answer your questions. Okay? Thank you very much for your attention.
I have a comment.
Yes?
A tooth being extracted and put in the vial. Is it also helpful if you use a syringe and suck up some of the goobers that are inside the site and
Yes.
throw that in?
Yes, it's helpful to let the socket bleed for 5 minutes even, and then suck it up. The longer you wait, the more microorganisms you will get. Because one of the things that Dr. Hal Huggins discovered was he at first he thought that the most toxic part was the root canal tooth itself. And then he discovered, no, the most toxic area is just outside the root canal. That's where the microorganisms were being fed best. And so that's where you get more microorganisms. It's that tissue just outside the root canal, which is why I have to add, if you're taking out a root canal, you must use a slow drill and drill out another 1 millimeter to 2 millimeters of bone that held the tooth. You have to remove all of the periodontal ligament. You have to remove another 1 millimeter to 2 millimeters of bone to make sure that you get a lot of those microorganisms out of it.
And Blanche, we have a question here where someone wants to know, is your lab considering testing for COVID?
We are considering the antibody testing, but we have not, we put in an application with the state of Colorado, and we have not heard from them yet. We don't do RNA testing, and that's what you need to determine the COVID virus. We don't do RNA testing, but we are capable of doing the antibody test. So we're waiting. Leslie is looking at it very carefully, and she's finding some faults with the test. And so we're certainly not going to jump into something that may not be 100% perfect.
What about the KBOM, that's the finger test?
I don't know anything about it.
Okay.
Leslie, do you?
Nope.
Okay. Nope.
Is that the finger prick test?
Yes.
Yeah, so they're looking for antibodies. There's like 20 or 30 tests that got fast tracked through the FDA, but they're finding a lot of faults with them, as in a huge percentage of false negatives, up to like 40%. So I'm just not sure if that's like a can of worms that we want to get into. It's not, we have the capability of doing antibody testing. However, it is not our specialty. So we would be utilizing a protocol supplied with a testing method, and to use someone else's protocol, we're a little hesitant to do that.
Okay.
Right.
Very
Okay.
good.
Any
All
other
right,
questions?
Blanche, I want to tell you it was another excellent meeting. I appreciate it. We're going to go ahead and end the recording so that this can be up tomorrow. And anybody who didn't
Right.
get to listen to it or wants to listen to it again or wants to share